Most people don't expect to be hard at work at age 74. Mary Del Chilton is not one of them. She loves science, and her pioneering work has already revolutionized the biosciences. It is also changing the way the world produces its food. That would satisfy most people, but Chilton wants to learn more, and she's pursuing that quest in a Syngenta lab, in a Syngenta building that are both named in her honor.
It's curiosity driven. I want to find out how things work. We use this kind of technology to analyze what makes a plant cell work. You know, what genes does it express and what do they do? What changes when we do this? So yeah, it's the technology is not just a tool for genetic engineering. It's a tool for the plant biologist to find out how things work.
That drive to understand how things work led to groundbreaking research on how a plant bacterium can be used as a tool to insert genes from other organisms into plant cells, and that could then produce crop varieties with new traits. Chilton and her team harnessed the gene transfer mechanism of the bacterium Agrobacterium.
So when we first discovered that Agrobacterium is putting novel DNA into plant cells, and somehow it was managing to stay there and replicate, I didn't believe it, you know. The first thing I wanted to do was prove that that was wrong. And in trying to prove that it was wrong, we found that it was true.
And that led to the production of the first transgenic plant. It also helped to create the field of genetic engineering of plants. Chilton won the World Food Prize in 2013 for her work. Now, 30 years after the discovery, dozens of companies, including Syngenta, are following in her footsteps, believing genetic engineering can protect plants from the environment, including pests and disease, and it can also increase the amount of a crop that farmers can produce.
By doing this biotech approach, you have always turned on protection there. So that's the power of biotechnology.
Take the corn rootworm. An infestation of this tiny bug can devastate hundreds of acres of corn.
So if you take a look, those are the newly hatched insects we call neonate.
Dr. Eric Chen is raising the worms in this specially designed bug nursery for his research. Dr. Chen and his colleagues have genetically modified corn plants with a trait that will kill rootworms.
We call it trait gene. So that's a piece of a gene or a DNA that we engineered in the lab, and then identify the activity that will kill corn rootworm, and then we'll put it into plant, integrate it into plant genome. So when the farmer buys the seeds, the trait gene's already in it. So what the farmer need to do is grow the seeds.
That's right. By changing the plant's DNA, researchers have created corn that will express a protein in every cell of the corn plant that will kill a rootworm soon after the worm nibbles a few bites of the plant. That effectively stops an infestation before it gets out of control.
This is the protein. This is the protease recognition site. So this is basically just 4 amino acids that a protease in the rootworm gut can recognize and then clip this protein right there. That activates it. Now it can bind to the rootworm gut and form pores, killing the rootworm.
Dr. Hart says her work helps farmers because using genetically modified crops is a more precise and cost-effective way to deal with pests than spraying fields with chemicals. It also lasts all season.
Buy the seed up front and then you have good protection. It's very consistent. You don't have to worry about anything washing away or diffusing in the soil. Your protection is inside the plant. Farmers definitely have the choice to go in and spray if they would like to. This gives farmers another choice. If they don't want to use the chemicals or they don't want to spray, they have a GM solution as well.
It takes almost 15 years to get a product from when it is first conceived to when it goes on sale, and almost half that time is spent in product safety testing. Researchers have to find whether the product is safe to eat and whether it's safe for the environment. Syngenta tests its potential new genetically modified seeds in this high-tech greenhouse in Research Triangle Park. It's one of the largest greenhouse facilities in the world. There are 30 climate-controlled areas, so the environment in Kansas can be simulated in one room with Africa in the next.
Scientists will take a tissue sample from leaves, from roots, from stalk, from ear, to make sure the gene is expressed in the right tissue at the right time with the expected level of expression.
If the analysis of plants grown in the greenhouse shows a single intact copy of DNA, meaning the genetic transfer was successful, the plant is moved to field testing to see if it's performing the way it should.
Then we'll do basic characterization studies so that we completely understand what went in, where it went in. We'll do studies in the field to say, does it grow like other conventional varieties of the same plant? Does it have the same composition in terms of fats, proteins, carbohydrates, amino acids, lipids, etc.?
Researchers want to understand how the genetically modified plant compares with the conventional variety of the crop. That includes similarities and differences. Scientists say every phase of the plant's life cycle, the part it plays in the environment, and its potential use is studied.
And so we'll do a series of studies to evaluate the impact of that difference to the safety of food and feed that may be derived from that plant, or to the environment if that plant is cultivated in the environment.
When we make decisions about technological products, often we think safety, benefit, and those are really the only two parameters that should matter. And then we take a step back and say, well, safety is based on science, but it's not always based on science. It's a shared notion of where we draw the line. What's safe to me may not be safe to you. And where our tolerance as a society is for these types of products is much more broadly based on risk, benefit, trust, our cultural worldviews, and other factors, even gender and race.
But despite the safety assurances, genetically modified crops have become a lightning rod for those concerned about food safety and environmental issues.
As scientists, we also always have to be doubting what we're doing and testing and retesting, and also trying to figure out why our brains, are so limited in what we can see. So that's always the challenge. And I think people have seen these new technologies, again, as I said, come and go, and here's a new one coming along. And not only that, but this one has two ingredients that make people, I think, a little nervous. One, it's their food. It's what they're eating. And the other part is it's genetics. And genetics has a long history of very, strong hot button issues with people.
The debate surrounding genetically modified crops prompted NC State to create a program in genetic engineering and society. Researchers will examine the cultural, policy, and economic aspects of genetically modified organisms. An estimated 17 million farmers around the world grow genetically enhanced crops, primarily in the United States, South America, and Asia. But much of Europe and Africa have blocked their use.
Genetically modified organisms appear to be falling through the cracks. Either they're entirely prohibited, as is the case with certain ones, or they are essentially unregulated. That's the great fear. And so there's this interesting moment now where folks on almost every side of the discussion are calling for some kind of regulatory reform.
Back at her lab, Mary Del Chilton is aware of the debate, but focuses instead on her discovery's potential, feeding the world. And she continues that work, trying to figure out the best way to direct a selected genetic trait to a specific place in the DNA sequence of a plant.
My task often is working with the biggest molecules. It turns out that I'm good at that. I'm better than other people. More experience. It has kind of a fingerprint that we look at to see if it's the right stuff. And if it is, we say yippee, and then we cut it again and stick the next piece in.